Hello there, this is Rohan Meshram again, and in our previous few series of videos, in our previous few series of lecture, we have dealt in more details of parasitic diseases. We have dealt with four parasitic diseases like we have dealt with Plasmodium, we have dealt with Leishmania, we have dealt with Filaria, and we have also studied Trypanosoma in more details. But now onward, we would shift our focus from parasitic disease to another class of disease called as bacterial disease. And to study bacterial diseases in more details, I have selected our model as Mycobacterium tuberculosis. So we would study the bacterial diseases taking this Mycobacterium tuberculosis as one of the model organism. Okay, to begin with, I would let you know about the introduction. Of course, we would discuss epidemiology, what is primary infection, what is latent infection, how these latent infections are reactivated, and what are its clinical manifestation. We would also talk about diagnosis and treatment of this tuberculosis. Okay, so just to give you a perspective, there are a lot of famous people who are diagnosed with TB and who are said to be TB survivors. Okay, so one of the most important person that you might know is that Mr. Amitabh Bachchan. He is a TB survivor. He was diagnosed with spinal TB back in 2000 while shooting Kaun Banega Crorepati. Another few people that you might be interested in was the cruel king of England, Henry VII. He was also diagnosed with TB back then. One of the most important scientific personality that eventually or unfortunately died with TB was Srinivasa Ramanujan. You might have seen the movie The Man Who Knew Infinity. In it, you might see him die with TB. Okay, anyhow, now let us continue with our discussion on biology of TB. So, well, let us first define what is TB and what exactly is this Mycobacterium tuberculosis. Okay, so this Mycobacterium tuberculosis, in short, we would refer it as MTB, is a bacterium that causes the disease tuberculosis. So if you look that Mycobacterium tuberculosis bacteria in microscope, you would observe it as a slender and rod shaped bacteria. This Mycobacterium tuberculosis bacterium is strictly aerobic. That means it would need oxygen to survive. And one of the most important feature of this bacterium is that it possesses a waxy cell wall, and it is this mycolic acid, the presence of mycolic acid makes the cell wall of this bacterium waxy. Okay, and it is due to this mycolic acid that makes the cell wall acid fast. Okay, so what do we exactly mean by acid fast? It is that it would resist the decolorization step in the training in the staining procedure that we usually use to identify gram positive and gram negative bacteria. Okay, so in that staining procedure, your Mycobacterium tuberculosis does might look as bright red in color. Okay, as you can see over here, this is a bright red on Zil Nielsen stain. Okay, so now this due to this waxy cell wall that is attributed to mycolic acid, this bacteria can sustain very harsh environment out even on dry surfaces for even months. So so this is a very resistant bacteria. It is very persistent. It won't die off very quickly, since it would survive on even dry surface for even months or weeks as such. Okay, so well now the very typical characteristic of this mycobacterium tuberculosis is that it is an intracellular pathogen of mammalian respiratory system. Okay, so it prefers to first infect the respiratory system, and it is known to cause what we call it as the granuloma formation. We are going to talk about more in details of this granuloma in upcoming few slides, but for time being, you should know that it is the bacterium that would cause granuloma to form in the lungs. Okay, so now, well, let us talk about how this disease or this bacteria is transmitted. Okay, the transmission of this bacteria is through droplet infection mode. Okay, so droplets are nothing but small particles of, we'll say, 1 to 5 micrometer in size, and each droplet of an infected person can contain about 1 to 400 bacilli, depending on what is the stage of infection that person is suffering from. Okay, so now each droplet thus can contain some 400, around 400 bacilli, and these droplets are usually expelled by person when he sneezes, or these droplets can be expelled during coughing, even laughing and singing. So once these droplet are present in air, and the size of this droplet is so small that they can remain suspended in air for even several hours. Okay, and unfortunately, if another healthy person pass out through that particular space during that time, these droplets can be inhaled by another person, and thus thus the bacteria can get entry into another healthy person's lung, and in this way the disease is transmitted from one person to another person. The most important factor that is to be noted is that unlike the previous diseases that we have learned, in case of plasmodium or leishmania or filariasis or your trypanosoma, these bacterial infection do not require vectors. Okay, so they are transmitted from one person to another person. In case of our in case of our Mycobacterium tuberculosis, it is spread through droplet infection. Okay, now, so once a healthy person inhales the droplet, these droplet now enters in the upper respiratory system. Okay, and the bacterium that is present inside those droplets get trapped in the mucus layer lining the epithelium of the upper airways of the lung. So here is the upper airway, what we call it as the upper airway of our respiratory system, and here is the detailed, or you can say microscopic view of this upper airway. As you can see over here, the epithelial cell that are lining these upper airways are surrounded by the mucus layer, and your bacteria, or not only Mycobacterium tuberculosis, but any of the foreign body or any of the foreign bacteria that gets entry through the upper respiratory system get usually trapped in this mucus layer. So what happens further is that once it reaches the mucus, once it gets trapped into this mucus layer, well, this mucus layer and the epithelial membrane do contain a small hair-like structure. We call them as cilia. Now, the cilia are supposed to be lining the surface of those epithelial cell, and the cilia moves together in a very coordinated fashion. They move together in a single direction or the other way around. Okay, so due to movement of this coordinated movement of the cilia in a single direction, that results in sweeping of those bacilli bearing mucus up towards the mouth, and the bacilli or any foreign particle or your mycobacterium, even in this case, can be possibly thrown out by coughing or are swallowed, rendering the host harmless. Okay, so this is the primary mechanism by which the respiratory infections are, are avoided by humans as such, by human body as such. Okay, but unfortunately, some of the bacilli do manages to dodge this system, and they make their way back into the lungs. Okay, so these bacilli might manage, or they get past to the upper airways defenses, and they finally reach to the alveoli. So these alveoli collectively are called, these alveoli is made up of a lot of alveolus. So alveolus, you can say, is the smallest functional unit of respiratory system, just like neurons are the smallest unit of your nervous system. Nephrons are smallest unit of your excretory system or kidney system, you can say. In the similar way, alveoli are the smallest functional unit of the respiratory system. Okay, so once it reaches the alveoli, the patient is said to have primary infection. As you can see in this lung, it has passed the upper respiratory tract and it has somehow reached to the alveoli. It is present over here. So it would be better now that I briefly explain you how the respiratory system of human beings are, so that it would be more clear for you to understand where our bacteria goes and what happens where. Okay, so let us now try to understand the anatomy of our respiratory system first. Okay, so our respiratory system is mainly composed of lungs and trachea. Okay, and our lungs has got we have got two pairs of lung. We call it as the right lung and left lung. Your right lung is made up of three lobes: superior lobe, middle lobe, and inferior lobe, while your left lung is made up of only two lobes, the inferior lobe and superior lobe. Now, due to presence of this cardiac notch, it might appear that the size of one lung might be bigger than the another lung. Okay, so what happens now is that you have got another component, trachea. This trachea, when it reaches to your left and right lung, it gets divided into two main primary bronchus. Okay, so these primary bronchus are further subdivided into many lobular or secondary bronchus, as you can see over here. And these lobular or secondary bronchus are further proliferated into tertiary bronchus, or what we call them as segmental bronchus. Okay, so this is how your respiratory system is. Well, now let us go into more details, or let us zoom into this anatomy of respiratory system at tissue and physiological level. Okay, so now here we are now in your tertiary level of of your respiratory system. If you zoom into the tertiary level, you might see that you have got this alveolus. Alveolus is present over here, and it is a collection of small sacs, which we call it as alveolar unit. Okay, and this is the alveolus where the oxygen transport occurs. Okay, so this alveolar unit is usually consist of cluster of sacs that are attached to a small airway, and this small airway is called as bronchiole. Now, we have got at least 350 million alveoli in each human lungs. So you can get the very complexity of how densely these alveolar units are packed in your lungs. Okay, so now let us again zoom into the alveolar unit, and as you can see in this figure, if we take a transverse section of this alveolar unit, you can see that there are these blood vessels, and these blood vessels are kind of proliferated around these alveolus that enables the efficient oxygen transfer. Okay, so now this blood capillary surround the alveolus, and this alveolus allows the exchange of oxygen and carbon dioxide molecules in and out. Okay, so now coming back to our infection of Mycobacterium tuberculosis, once these mycobacteria, after escaping the host defense system of upper airways, and they move down to the bronchiole and to the individual alveoli, we say that the person has got primary infection. Now, this MTB is an obligate aerobe. It thrives in oxygen-rich environment. Thus, these alveoli of human lungs are very ideal location for these guys to settle in. Okay, so since lungs are the places where most of the aeration happens, so and your mycobacterium may require uh, a lot of oxygen. Therefore, we can say that it is an ideal location for them to thrive. Okay, so